Hello, my name is Lindsay Reynolds, and I am the Riparian and Wetland AIM Lead with the Bureau of Land Management at the National Operations Center. The following presentation will cover field methods for annual use in Section 8 of the Field Protocol for Lentic Riparian and Wetland Systems. Why do BLM monitoring projects collect annual use data? We collect annual use to monitor livestock, wildlife, and human use at a site. Annual use data can help determine whether current grazing is meeting criteria. It can serve as an early warning indicator for management and condition. Annual use data helps inform permit renewals, and annual use data helps meet compliance. There are 3 methods protocols included in the annual use section, which I will cover in this presentation. Section 8.1, stubble height, Section 8.2, soil alteration, and Section 8.3, woody use. The materials needed to conduct annual use sampling are the woody structure and annual use datasheet, or the annual use only datasheet, a clipboard and pencil or an electronic device for paperless data collection, which is preferred, a graduated survey rod or height measuring stick with graduations in centimeters or meters, such as an avalanche pole with clear markings, a MIM frame, which is the eye-shaped quadrat frame shown in the top middle of this slide, which is 50 centimeters long by 42 centimeters tall, and typically made of half-inch PVC pipes and couplers. You'll need 3 30-meter measuring tapes and chaining pins to secure the transects. The first annual use method I will cover is the stubble height method in Section 8.1. To begin the stubble height method, you must first establish and anchor the transect. Then, step 1 of the methods is to complete the top section of the datasheet, or the first page of the Survey 123 form, with information about the site, the observers, and recorders. Step 2, the data are collected at 1.5 meter intervals along the transect in a 50 centimeter by 20 centimeter quadrat shown by this diagram on the right. The first point for data collection is at the 0 meter mark on the transect tape. At each designated mark, place the MIM frame along the transect, as shown in the diagram, on the opposite side of the tape from where you're standing to form a 20 centimeter by 50 centimeter quadrat. The long 50 centimeter edge of the MIM frame should be parallel to the tape, and the left short 42 centimeter edge should be perpendicular to the tape in line with the designated mark. The quadrat for stubble height is the half of the MIM frame closest to the tape. Step 3 is to locate the graminoid, or key species for measurement, a 7.5 centimeter tuft, closest to the upper left corner of the quadrat formed by the MIM frame and the transect tape, and shown in this diagram with the black circle. In this protocol, the default is to measure stubble height on the closest graminoid vegetation within the measurement area. Stubble height may also be measured on specific key species that are identified by the monitoring objectives of a site or project. Make sure to check with your project lead to determine whether stubble height will be measured on all graminoids or specific key species. Most riparian graminoids grow tightly together, forming dense mats with little separation between individual plants. Often, several rhizomatous species may be growing together. Thus, the method uses the 7.5 centimeter diameter tuft of all co-occurring graminoid vegetation. If graminoid vegetation, or key species, are sparse and part of the closest tuft is outside the MIM frame, measure the entire 7.5 centimeter tuft. If graminoid vegetation, or key species, do not occur as a homogeneous 7.5 centimeter diameter tuft anywhere in the frame, but as a smaller individual plant less than 7.5 centimeters in diameter, measuring a smaller tuft of leaves closest to the corner is acceptable. Stubble height is measured for graminoid species only. if the vegetation in the frame is a mix of graminoids and forbs, remove the forbs from the tuft before measuring. Once you've identified your tuft of graminoid vegetation, step 4 is, using the measuring rod or a separate ruler, identify the dominant or key species within the tuft and measure the average length in centimeters of all the graminoid leaves. For each tuft, record the USDA plant species code or unknown code of the dominant or key species in the species column of the datasheet. If there are multiple species intermingled in the tuft, record one species that is dominant in the tuft, but measure the average leaf length for the whole multi-species tuft. Next, determine if the tuft has been grazed. Mark a yes in the graze column if it appears that any leaves in the tuft have been grazed. Otherwise, mark no. Next, record the average leaf height in the stubble height column. Measure the leaf height only, do not measure seed stalks or combs. Determining the average leaf height will take some practice. Be sure to include all leaves within the tuft. The easiest method of doing this is to grasp the tuft, stand the leaves upright, and then measure the average leaf height as shown in the photos on the right. All leaves, grazed and ungrazed, are measured from the ground surface to the top of the remaining leaves. All leaves within the tuft should be lifted to determine their length. Account for very short leaves as well as tall leaves. Here is an example of how stubble height would be recorded on a datasheet. For each location on the transect, in this case, the 0 meter location, the dominant graminoid species in the tuft grab is recorded, in this case, Elymus canadensis, or ELCA. Then it is marked yes or no whether it is grazed at all, in this case, yes. Then its leaf height is recorded in centimeters. In this case, the average leaf height of the tuft grab of Elymus canadensis is 40 centimeters. Here's an example of what the electronic data form in Survey 123 looks like for stubble height, showing information for a tuft grab of Elymus canadensis, a stubble leaf height of 40 centimeters, and yes, it is grazed. There are several measures you can take to ensure quality data collection. These quality assurance steps for stubble height are also listed in the protocol at the end of section 8.1. Please review these steps during and after a sample event. The next annual use method in section 8 of the field protocol is soil alteration, section 8.2. To begin the soil alteration methods, you must first establish and anchor the transect. Step 1, the data are collected at 1.5 meter intervals along the transect in the same quadrat as stubble height, but this time using both sides of the membrane. So a 50 centimeter by 42 centimeter quadrat, shown by this diagram on the right. The first point for data collection is at the 0 meter mark on the transect tape. At each designated mark, place the membrane along the transect on the opposite side of the tape from where you're standing. The long 50 centimeter edge should be parallel to the tape, and the short 42 centimeter edge should be perpendicular to the tape in line with the designated mark. The quadrat for soil alteration includes both halves of the membrane. Step 2 is to count soil alteration within the quadrat. Mentally project 5 lines, 2 end bars of the frame, and 3 intermediate lines spaced 12.5 centimeters apart, across the frame perpendicular to the transect and the center bar of the frame, as shown with the red lines in the diagram. Then count the lines that intersect soil alteration. Look at the entire quadrat, which includes both halves of the membrane. Let's talk about what counts as soil alteration on the soil surface. There are detailed steps in step 2 of the field protocol. The soil surface is considered altered when one or more of the following are present: hoof prints from livestock, wild horse or burro, or native ungulates, human footprints or wheel or tread tracks that depress the soil and expose bare soil at least 13 millimeters or half an inch deep. A vertical face that has formed from hoof shear, such as in the photos to the right. Displaced soil is moved into a pile that's at least 13 millimeters or a half inch high. Or compacted soil or an unvegetated path caused by repeated movement of hooves, feet, wheels, treads over the same area, for example, a livestock, game, or foot trails, or a vehicle path, even if the soil depressions may be less than 13 millimeters. Finally, count only the current year's soil alteration. Trampling impacts must be obvious results of the current season use. Old features tend to be non-distinct. Obvious soil alterations are defined as those that are readily observed from no closer than approximately 2 feet from the ground surface. In general, these are impacts that are evident without kneeling close to or lying on the ground. When considering paths or trails, trails that experience use during the current season and remain unvegetated are counted. Pre-existing trails that have revegetated are not counted. Finally, step 3 is record the number of lines from 0 to 5 that intersect soil alteration. Record only one occurrence of alteration, trampling, compaction, puddling, or hoof shear per line, even if more than one occurs along the line. If a single feature intercepts more than one line, record one occurrence for each line. Record a 0 for no alteration. Do not leave the cell blank if there are no alterations. Here's an example of how soil alteration would be recorded on a datasheet, showing data recorded at the 0 meter location on the transect, and 2 lines in the memframe were intercepted with soil alteration, so a 2 is recorded in the far right cell for that line. Here's an example of what the electronic data form in Survey 123 looks like for soil alteration, showing that 2 lines in the memframe were intercepted with soil alteration, so a 2 is recorded in the soil alteration line. There are several measures you can take to ensure quality data collection. These quality assurance steps for soil alteration are also listed in the protocol at the end of section 8.2. Please review these steps during and after a sample event. The final annual use method in section 8 of the field protocol is riparian woody use in section 8.3. To begin the riparian woody use methods, you must first establish and anchor the transect. Step 1. Measure riparian woody use at 1.5 meter intervals along the transect in a 2 meter by half meter quadrat. The quadrat for measuring riparian woody use is formed by 1 meter on both sides of the transect and 50 centimeters along the transect tape, beginning at the 0 meter end and repeated every 1.5 meters thereafter, as shown in this diagram. This is the same quadrat as woody structure methods in section 6.4. Mentally project a 3-dimensional box through the vegetation around the 2 meter by half meter quadrat and extending to 2 meters in height. This is the area for assessing woody use. Step 2 is to identify all riparian woody individuals rooted in or overhanging the 2 meter by half meter quadrat. In this protocol, the default is to measure only riparian individuals for woody use, so only individuals that are designated as FAC, FAC-wet, or obligate wetland species. Riparian woody use may be measured on specific woody key species that are identified by the monitoring objectives of a site or project. Make sure to check with your project lead to determine whether riparian woody use will be measured on riparian species or specific key species. In step 2, you record the location in meters along the transect associated with the quadrat in the location column, for example 0, 1.5, or 3 meters, etc. Since there may be multiple woody individuals rooted in the plot, There may be multiple lines on the datasheet for each location. Each line must have an associated location in the location column. Next, for each individual, record the USDA plants code or unknown code in the woody species column. For example, shown in the diagram, we have 5 woody individuals represented by light green stars that are either rooted in or overhanging the quadrat. Each of these 5 individual species names would be recorded on its own line in the data sheet. Step 3 is, for each individual, determine the available current year's leaders that intercept the quadrat at a height of 2 meters or lower. So, for each individual, determine how much of the current year's leaders, or current year's growth, in other words, new branches, are available. Growth is available for grazing or browsing if it occurs below 2 meters, or 6.5 feet, in height, which is considered within reach of most browsing animals. If the individual's leaders are entirely above the reach of the grazing animal, or above 2 meters, they're considered unavailable for browsing, and those leaders are not assessed for riparian woody species use. If all leaders of a given riparian woody species are unavailable, do not record use for that individual. Mark the individual as unavailable. Here are some examples of browsed willows, a riparian species. The rod shown in the pictures is 1.5 meters high for scale. In the photo on the left, most of those leaders are below 2 meters, so most of them are available. There is some browse on the lower portion of the willow. You can see leaves missing, so the rating for this individual would be slight or light. In the photo on the right, there's actually another willow in the foreground that the rod is leaning against. You can see that it's all lower than 1.5 meters, so it's all available. And most of those branches have been browsed and stripped of their leaves, so the browsing on the right individual would be heavy or severe. Here's another example of browsing on some willows. There are 2 willows shown in the photo, and outlined in green here. These are low willows, both less than 2 meters tall, and you can see the willow on the right has not been browsed at all. The leaves are all intact, but the willow on the left has been browsed quite aggressively, with almost all leaves stripped off. The last step in evaluating woody use, step 4, is to determine the woody species use class for each individual. So, the use class of individuals' leaders that intercept the quadrat at a height of 2 meters or lower. Plants are classified into a use class depending on the degree of use within the woody browse quadrat. So, the 2 meter by half meter by 2 meter high box over the quadrat. For each individual, record the woody species use class on the datasheet using the midpoint of the use class in Table 14. Repeat the use class determination for each riparian woody species individual encountered in the quadrat. In the example diagram, record use for each of the 5 individuals shown in the quadrat. Review use class descriptions periodically while reading the quadrats to maintain precision and accuracy. Here is Table 14 from the field protocol, the woody species use classes and descriptions. The use classes range in 20% increments, and the midpoint of each class is recorded for each individual. For example, the light use class is 21 to 40% use on an individual, and you would record 10 for slight use, which is the midpoint. Here's an example of how riparian woody use would be recorded on a datasheet, showing data recorded at the 1.5 meter location on the transect. In this example, there are 5 woody individuals, but only 2 of them are available. The Ribes aureum, Riau, which is marked as a use class 10, or slight, and one of the Salix goodingi, Sago, which is marked as a use class 50, or moderate. You can see the riparian woody use class data are recorded on the far right cells for each riparian woody individual. In this case, the Tamarus gramosissima, Tara, is dead. The name has been circled, so we don't record use for it. And the last two individuals, a Populus fremontii, Popher, and another sago, both have branches above 2 meters and are not available. They are marked unavailable. Here's an example of what the electronic data form in Survey 123 looks like for riparian woody use, showing a sago with a use class of 10 or slight. There are several measures you can take to ensure quality data collection. These quality assurance steps for riparian woody use are also listed in the protocol at the end of section 8.3. Please review these steps during and after a sample event. The annual use methods can be collected alongside of the core methods in a second pass of the transect after LPI data are collected. If annual use are being collected with core methods, they are collected at the same time as woody structure, which is also collected at 1.5 meter increments along the sample transect. Here in this diagram, you can see how, in this case, you would lay the MIM frame down in the woody structure in use quadrat, measure the stubble height and soil alteration, and then measure woody structure on any woody individuals in the large quadrat, and use on any riparian woody individuals. Then you would continue down the transect to the next 1.5 meter increment, lay the MIM frame down for stubble height, measure soil alteration, and then record woody structure and use and continue down the transect. In this way, annual use can be recorded along with the core method's woody structure in a second pass of the transect. This concludes the presentation for annual use methods in Section 8 of the Field Protocol for Lentic Riparian and Wetland Systems. Thank you for listening.